And welcome, Biomanians. Yeah, that's who you are. You are the people who are here on a Thursday night. It is, of course, 1 week before the AP Bio exam. In fact, 1 week from tonight, it'll be all over. It'll be all over. But nevertheless, I can't tell you how much I admire those of you who are here watching. It is really cool to be here with you tonight. We are broadcasting from learn-biology.com world headquarters. That's in Berkeley, California. And it is just so great to have all of you here. So really fun to be here tonight. Beautiful day in Berkeley, beautiful day in Berkeley, California today. And I hope your weather was good around the country or even around the world. I've had Isaac actually already asked me a question about operons and about the phospholipid bilayer and electron gradients and pumps, and that's fantastic. We can definitely do that. And if anybody else has any other questions, you can just chat in, use the comment feature on YouTube. And again, I'm so glad to have all of you here. The agenda for tonight originally was to focus on Unit 2. We did Unit 1 in our last session. And to also, by request, do some FRQs. So I'm ready to do that. We can do some of the FRQs that are on learn-biology.com. We can do some of the FRQs that are on the College Board's website. So anyhow, it's all good. And let's go. I'm going to go like this and nice. And I'm going to go to the next slide, which I'm going to go like that. So tonight's agenda. Again, want to welcome you. You are Biomanians, and I'm a Biomanian too. Biomanians are the folks who just love BIOLOGY. And sometimes Biomanians need to put themselves in a different place on the screen, like I'm going to do right now. That's great. And plan for tonight was to review Unit 2, as I said, to do some FRQ review and to do some MCQ review if we've got time. So again, really glad to have you all here tonight to learn some BIOLOGY. I usually introduce myself at the top of the show. I'm Glenn Wolkenfeld, also known as Mr. W. I'm a retired AP biology teacher, but this year I taught more biology than pretty much any other year. At least I worked harder at it. And I'm very grateful to be able to do this and to be able to teach people around the world through YouTube. It's pretty fantastic. I want to give you a couple of tips about some moves for success. And the first move for success is I've got a checklist. AP Bio Review should be a process of self-assessment and then filling the gaps. Self-assess, fill in the gaps. And I put together a checklist that runs you through the entire course, and you should download it. It's at apbiosuccess.com/checklist. And you just, put your email address in, bam, I email you the checklist. And it's super useful in terms of assessing where you are. And start with that. In addition, I just want to tell you, AP Bio exam, less than a week away. You want to study in the most efficient and effective way. And the most efficient and effective way is to do that with interaction and feedback. And that's why I wrote learn-biology.com. It's the website that guarantees success. And it guarantees success because it tells you what you know and what you don't know. It interacts with you. It's like having a 24/7 tutor available to you all the time. Right now, you should be assessing with the checklist and then going on to learn-biology.com and just working on questions. Have your study be active. That's the mistake that many students make as they study for an exam like this. They do passive things. They reread their notes. They watch YouTube videos. Now, I mean, guilty as charged. I've been putting up YouTube videos, and I want you to watch them. But after you watch those videos, I want you to go to learn-biology.com, and then I want you to practice in an interactive way. I don't want to pretend that you couldn't do it on your own. You can do things like make flashcards, but why should you make the flashcards when I've already made them for you? 470 something flashcards on the site. So again, make it easy for yourself. The site is not free. I'm here to encourage you to buy it. It's $24.95. It's a monthly subscription, and you can cancel anytime, which means that your total cost will be $24.95. So I really want to encourage you to make the study easy for yourself. Make the move that's going to lead you to AP Biology success. And that move is to sign up for a subscription of learn-biology.com. I'm going to be saying that throughout the show, but I'd like to start letting you know what I think is your best move for success. We're going to review cells, but boy, Isaac, you wanted to go over the phospholipid and stuff like electron gradients and pumps. Isaac, did you specifically mean that like you wanted to review the electron transport chain, chemiosmosis, that kind of thing? Is that what you're interested in? Let me know in the comment thread. Operons, I love operons. I have a song. I mean, I love operons so much that I wrote a rap song about them just this year. So Isaac, electron transport chain. So let's see, what's an easy way to do that? Okay, let's just give a quick once over of the electron transport chain. I think that the easiest way for me to do that is to go like this and to go like this. And I'm just going to remember that I was like on slide 29. Okay, and what we're going to do is we're going to, oh, perfect. All right. So, Isaac, this is for you. This is for you. So, electron transport chain. Just want to start with reviewing what happened before the electron transport chain. Before the electron transport chain, we have processes like glycolysis, the link reaction, and Krebs. Glycolysis and Krebs make a little bit of ATP, but the main thing that they do is they make many of these mobile electron carriers. NADH, FADH2, those are mobile electron carriers. The way that I encourage students to think about them is they are like batteries that can have a charged state and an empty state, you know, a spent battery and a charged up battery. So they're rechargeable batteries. NADH is the charged form. FADH2 is the charged form. NAD plus and FAD, those are the spent forms. Those are the batteries that you have to put back into your rechargeable battery charger to get them to be full of energy. So in previous stages of cellular respiration, NAD plus was made into NADH. FAD was made into FADH2. Now, those molecules are in relatively high concentration in the matrix of the mitochondrion. Here's a mitochondrion over here. Here's the outer membrane. Here's the inner membrane. And this is the matrix. So these are in the mitochondrial matrix. And they don't know where to go. Things inside cells are moving randomly, but because they're in high concentration, they will make their way over by basically diffusion to the mitochondrial inner membrane. In the mitochondrial inner membrane, there is a series of connected enzymes, proteins that form the electron transport chain. And that electron transport chain is represented by this yellow arrow over here. Okay, it starts with this NADH over here. It ends over here with oxygen. So we have these charged batteries that are coming to the electron transport chain, NADH and FADH2. They're going to discharge their energy. When you put a battery into a device, that device does some work for you. Well, NADH and FADH2, they're batteries. They're powering a process. They're powering an electrical process. What process are they powering? They're powering the pumping of protons from the matrix to the intermembrane space. From the matrix to the intermembrane space. And that is an active transport process because things are going from low concentration to high concentration. So by active transport, we're pumping from low concentration to high concentration over here. And all of those protons are trapped inside the intermembrane space. The intermembrane space, like all membranes, is primarily a phospholipid bilayer. Phospholipids don't allow charged particles to pass through. So these protons need a way out. They need a way out for two reasons. One is that they're in high concentration here, and things like to move from high concentration to low concentration. The other thing is that these protons are positively charged, and they're next to all these other positively charged particles, and that's an unstable situation. Like charges repel. So these protons want to get out. They really want to get out of the intermembrane space. The only way that they can get out is this. It's number 6 over here. 6 is a channel, and it's an enzyme, and it's called ATP synthase. ATP synthase. And what it does is it allows those protons to diffuse from the intermembrane space back into the matrix. As they diffuse through, their kinetic energy is used to carry out a very endergonic reaction, and that is to get ADP and phosphate to become ATP. That's the whole story, except for one little piece. Oxygen over here. This is called oxidative phosphorylation. Oxidative phosphorylation. Why do we need oxygen? We need oxygen because oxygen is the final electron acceptor. So electrons are flowing down this electron transport chain. They have to go somewhere. Where they go is to oxygen. And as oxygen accepts those electrons, it also accepts protons, and oxygen becomes water. Oxygen becomes reduced to water. And that is, it's not really the reason why we breathe out water vapor. But there is like a rat that's called a kangaroo rat, lives in the desert. Most of its water is called metabolic water. It's made through this process over here. So Isaac, that was the electron transport chain, and I hope that did it. And good. So, and also I just want to emphasize, like, if you're interested, I'm gonna show this, because how can I not show this? It's part of the reason why I'm here. I'm going to go like this. I'm going to type in. Oh, good. Okay. So if you, Isaac, were to go to learn-biology.com, click on courses, click to AP Bio 2.0, and then just go to cellular energetics. I've got a complete tutorial about the electron transport chain in chemiosmosis. And I'm sure that what I did was helpful, but I wouldn't feel confident at this point until I've done some interaction. And you can do additional interaction by going here. Go to AP Bio Review, click Multiple Choice Review, click Start Quiz, click OK, and then you'd select cellular energetics. If you just wanted to do cellular respiration, you'd uncheck all of this. And I'm going to do that again, and bam. Okay. Hey, this is perfect. This is perfect. Let's do this right now. The figure below represents a biochemical pathway in a cell. In the figure, the 4 compounds labeled 1, 2, 3, and 4. Let's interact, guys. Here we are. Are they fat, ATP, water, carbon dioxide? Are they glucose, citrate, acetyl-CoA, lactate? Are they glucose, pyruvate, lactate, and acetyl-CoA? Are they starch, pyruvate, carbon dioxide, and acetyl-CoA? So I'd like you to put a number into the comment thread: 1, 2, 3, 4, and 5. 1, 2, 3, 4, and 5. Ha! Okay, I'm seeing all these questions, and I'll try to get to as many of them as possible. But folks, I just asked you a question. I want to see a 1, 2, 3, or a 4. I know there's a lot of latency on this YouTube platform, YouTube interactive platform, but nevertheless, I'm going to take a sip of water when I'm done. Okay, a lot of people are putting in 4, and I really appreciate that. That's what makes it fun. Someone named IG is putting in a 3, and that's pretty great. Now Natalie is putting in a 3, and Lily is putting in a 3. Hello, Lily. Lily is a subscriber. Lily, I hate to just, but you know, here we are. It's a public forum. Lily is a subscriber to learn-biology.com. I'm sure that Lily's going to get a 4 or 5 on the AP Bio exam. Let's try 3. Excellent. Glucose is 1. This would represent glycolysis. So that means this would be pyruvate. If there's no oxygen, pyruvate gets broken down into lactic acid so that glycolysis can continue. That's a fermentation. And then oxygen, and then in the presence of oxygen, this would become acetyl-CoA. Okay, so good job to the many of you who made the right choice. And you know, if you didn't make the right choice, there's so much time to get yourself to a very, very high level. Okay, I'm reviewing the comment thread. Okay, Natalie, I want you to narrow your question a little bit. Like I can do any aspect of natural selection, evolution, ecology. There's just so much there. So, I mean, do you want to know about speciation? Do you want to know about population genetics? You know, stuff like that. Just, Natalie, narrow your question and then ask me another question in the chat. Isaac said, thanks, that helps a lot. Shayamala, you said, can you please discuss how to know which type of graph to use when graphing different types of data for FRQs? You know what I am going to do? Let's see if I can find this. Samantha or Shyamala, there's a website and it's called, like, if you Google which graph to use, there's a graph that has a tutorial on this. It's a big topic, and I think that I'm better served like answering specific biology questions right now. I know that's a little disappointing, but I think we'll do better if we do that. So Shyamala, can you please do that? And Samantha, with Mr. Snyderman, yay, love Mr. Snyderman. Light-dependent and light-independent reactions during photosynthesis. I can sure do that. Let me check the chat. And Natalie, speciation and osmoregulation, freshwater fish. Okay, so many questions. And you know what? This is what I've been waiting to do the whole time, to just be here answering questions. So fun. So I'm going to do my best. We are going to talk about photosynthesis. And to do that, you're still looking at that. I'm going to go back to my slideshow. Hope goes back. Okay. I could always have gotten back there, just so that you know. Okay. So photosynthesis. Let's find some photosynthesis here. Okay. Yeah, good. Okay. Click the wrong button. There we go. Okay, fantastic. So this goes out to Samantha in Mr. Snyderman's class and down in Los Angeles, California. So we've got the light-dependent and the light-independent reactions. Light-dependent, you know, those are the light reactions. Light-independent reactions are also known as the Calvin cycles. So big picture on photosynthesis. Light is used in the light reactions by these structures that are called thylakoids to create ATP and NADPH. So that's what the light reactions are doing. They're churning out ATP. We already know what ATP is. That's the molecule that cells use for energy. NADPH is just like NADH. It's a mobile electron carrier. It's used to reduce things, to give them electrons. So that's what comes out of the light reactions, in addition to something else that's super important. For that process to occur, water is required as an input. Water gets split apart. Water gets oxidized during photosynthesis. And as water gets oxidized, it donates its electrons. Donates is a very kind way to say it, because really what's happening is water's electrons are getting ripped away from it. And as water does that, it loses its electrons, it loses its protons, and what comes out is oxygen. So that's like such a fundamental biology question, and that is certainly something that could be asked on the AP Bio exam as well. So just know that like oxygen is a waste product for photosynthesis, but you know that has had planet-causing implications, like it's created our oxygen-rich atmosphere upon which aerobic metabolism is based. So we're still at a very big picture, Samantha. And now what happens is that in the Calvin cycle, the products of light reactions, ATP and NADPH, are used to, and listen to how I say this, they're used to reduce carbon dioxide into carbohydrates. What does reduction mean? Reduction is when you give something electrons, all right? Oil rig, oxidation is loss, reduction is gain. Very counterintuitive, but that's how it works. So carbon dioxide receives electrons and protons, and on the other side of that, it comes out as a carbohydrate. So that's the big picture of photosynthesis. Just going to skip that. Okay. During the light reactions, what happens? There are two photosystems. In photosystem 2, it's the one that comes first. I didn't do that. I'm not responsible for that, but that's the way it's always been. They were named in the order of their discovery. In photosystem 2, what happens is that light strikes a molecule of chlorophyll. Chlorophyll is a pigment that can donate electrons. We don't have to go into all the details of that. I don't think you need to know them, but just know that chlorophyll winds up getting oxidized, gives up electrons, and those electrons flow through an electron transport chain, not unlike the one that we talked about for cellular respiration in response to Isaac's questions. And as that electron transport chain operates, it powers another proton pump. That proton pump pumps protons from the stroma into the thylakoid space. The stroma is like the cytoplasm of the chloroplast, and the thylakoid space is the space inside the thylakoids. So stroma to the thylakoid space. Here's a larger version of that, stroma to the thylakoid space. And it's exactly what I just explained. Is it exactly? I don't know if it's actually, I think, okay, here's the deal. ATP is the same. It's homologous between the two processes. It's one of these ancient molecules that all of life is really dependent upon. I don't really know if this structure, if you'd call it analogous or homologous, and we're not going to worry about that right now. But in the same way as protons were trapped in the intermembrane space of a mitochondrion, they're trapped inside the thylakoid space of the thylakoids in chloroplasts. They can only get out through one channel, and it's a little bit bigger over here. All right, so you know this, same as this. So here's the protons that have accumulated here. They can only get out through ATP synthase, and as they do, ADP and phosphate are converted into ATP. Again, a hugely endergonic reaction, but it's powered by the diffusion of protons from the thylakoid space to the back to the stroma. Okay, so I hope everybody's clear on that. Now, one or two last pieces. Another thing that happens in photosystem 2 is that photosystem 2 has a water splitting complex, a bunch of proteins that can split apart a water molecule. We talked about that a minute ago. When that water molecule gets split, oxygen gets released, but protons also accumulate. It increases the gradient of protons over here, and that means more ATP is going to be synthesized. Now, we're almost done with the light reactions. The electron had done all this work powering proton pumping over here. It arrives at photosystem 1 depleted, out of energy, but light is still shining on the plant. As light shines on photosystem 1, that electron gets boosted over here and it goes into another electron transport chain. This electron transport chain goes to an enzyme that's called NADP plus reductase. What does that enzyme do? The name tells you. It reduces NADP plus into NADPH. Now we have the second product of the light reactions. We've created ATP and we've created NADPH. And that was a lot of talking on my part. And I just got water on my glasses. Not a good move. Okay. Better. Okay, so that, Samantha, was the light reactions. The Calvin cycle. In the Calvin cycle, what happens is that an enzyme called rubisco takes a 5-carbon molecule called RUBP and jams a carbon dioxide onto it. That's the first part of the Calvin cycle, and that's called carbon fixation. That pulls carbon from the atmosphere into the biosphere, into organic compounds. And right now, touch your arm, just like I did, okay? Every single carbon atom in my arm went through this process. You were once, or the carbon atoms that made you up, were once carbon atoms floating as a part of carbon dioxide in the atmosphere, and through the Calvin cycle, they got pulled into the biosphere, basically. Now, phase 1, carbon fixation. Phase 2 is called energy investment and harvest. The products of the light reaction, ATP and NADPH, are used to energize this molecule. They give it a phosphate group, they give it electrons and protons. At the end, what you have is G3P, glyceraldehyde 3-phosphate, and that can be harvested from the cycle to make carbohydrates, and those carbohydrates are used for the plant to grow. So we've done carbon fixation, we've done energy investment and harvest, and now the only thing left to do is rebuild RUBP. And there's a variety of enzymes that participate in taking this 3-carbon compound, G3P, and reconfiguring it so that at the end, it's this molecule, RUBP, and it takes a little bit of ATP also from the light reactions to make that happen. So, Samantha, how was that? That was the whole thing. That was the Calvin cycle. And again, I just got to show you guys, if you're not already subscribing to learn-biology.com and you're thinking, oh, it's too late, excuse me, but that's not correct. Let's go there. Go to learn-biology.com, and again, go to AP Bio version 2.0. If you were feeling like Samantha was feeling, oh, I don't really know that much about photosynthesis, bam, it's all right here. Interactive tutorials. In fact, light reactions, you would read about this diagram, you'd read about chlorophyll, you'd watch a video, and then you would do a quiz. All right, I'm going to get there. You do a quiz. And so the quiz would be like this. And the first part of the quiz, you'd have to label all the parts. Now, again, think about how much you would learn by doing this. It's phenomenal. And, you know, like where's the stroma? And let's say that you got it wrong. Let's say you put it here. You'd be told it was wrong. So, okay. Oh, yeah, I remember. Stroma's over there. So you get the kind of feedback that you need to really understand these monstrously complex processes. So again, I know that I'm saying it, but this is one of the reasons why I'm doing this show. I want you to buy learn-biology.com. I really want you to. Why do I want you to do it? Well, obviously I have a business and I'm selling it. But in addition to that, it is the #1 thing that you can do to get yourself to that 4 or 5 level. And people like Samantha don't need to worry about that because she's part of a class where she's, her teacher is using it. But if that's not you, you're competing against 8,000 people who are using learn-biology.com, and you should get on board. And over this weekend, you should be spending the entire weekend on learn-biology.com and also writing FRQs. Like, do those 2 things, and you'll set yourself up for a 4 or a 5. So, Samantha, how was that? Did that do the trick? Good. Okay, Samantha, good to hear. Natalie, origin of life. Okay, RNA hypothesis. Yeah, I can definitely do that. And Natalie, since I think I didn't take your first question, yeah, let's do that. Okay, so I'm thinking about the best way to do origin of life. Okay, origin of life. Here's a couple of things you need to know. I'm going to do this by going to here, AP Bio 2.0. I'm going to do evolution. And I've got a tutorial about the origin of life. And I think here's really what you need to know about the origin of life. Okay. I can make that bigger. That's pretty good. Okay. So here's really what you need to know about the origin of life. The big idea about the origin of life is that a couple of things needed to happen in order for life to emerge. Think about life. It's made of all these organic compounds, and those organic compounds are all organized within cells. So how did those organic compounds come about? Now, with the presence of life on this planet, it's life that creates life. Like for example, we just saw in photosynthesis. Photosynthesis pulls in non-living matter, carbon dioxide, and it makes it into living matter, carbohydrates, the fuel of life. How do you get stuff like that to happen in the absence of life? That's the tricky thing. So the first question is, how do you abiotically create monomers? How do you create the building blocks of the molecules of living things? What are we talking about? We're talking about things like monosaccharides, amino acids, nucleotides. How do you get all those building block molecules to emerge without life? And the basic idea is that it has to happen. There's no biology. So it had to happen through some kind of geological process. And I'll explain one in a minute. Then those monomers have to combine and form polymers. And then the whole thing needs to get organized inside cells. Was this an easy process? No. Did it happen quickly? Well, yes and no. There's indication of life on planet Earth 3.8 billion years ago, a really long time. But that means that there was about 100 million years after the Earth had kind of cooled and it wasn't being hit by massive asteroids. 100 million years for multiple processes to be happening all over the planet, and it only had to happen in one place. And we know that it only happened in one place because all living things are deeply interconnected. It's not like life arose twice. Life arose once. And what did that living thing have? That living thing had, and this is actually something everybody here should know, this could be an FRQ. All living things are unified by DNA as the genetic material, ribosomes to do protein synthesis, a membrane that's created out of something that's either a phospholipid or something very much like a phospholipid, RNA to transfer information from the DNA to the ribosomes. Chemiosmosis happens in all living things. So this process that I just talked about. So like that list is something you should be able to generate. It's very, very important. Like that's really hot material for an FRQ. So whose question am I asking? Natalie. So Natalie, those are the basic steps. Now, in the 1950s, there was an experiment that was done that proved that abiotic synthesis, abiotic, without life, synthesis of the monomers of life was possible. Okay? It's not like life was created in a test tube. That's never happened. But you could create simple monomers. The guy who did it was named Stanley Miller. He did it when he was in his 20s or something like that. And basically, he working at, I think he was at Caltech. No, University of Chicago, sorry. He put together this experiment. So it's a closed system that was completely sterile. There was a simulated ocean that was this sort of body of water. The water was heated, boiled by a Bunsen burner or some other heating device. That went over to a simulated atmosphere, and the simulated atmosphere had lightning strikes going that were simulated by these electrodes in it. The atmosphere had a bunch of gases. They were methane, ammonia, hydrogen, and water vapor. And the reason why those gases were used was that the first atmospheres that were discovered were those on planets like Jupiter. And that is a very friendly atmosphere for this kind of experiment. But this experiment has been rerun thousands and thousands of times. So the idea is that water was running through the system, water vapor, there's electricity coming over here. And Stanley Miller let this run for a couple of days or weeks, and amino acids were found in this trap over here. So amino acids spontaneously formed with a certain amount of energy. Now, it's actually unlikely that this is the way that life's monomers originally formed, but it was something, some abiotic, some non-living process gave rise to life. And that enabled us to get monomers. There are other processes that lead to polymers, and that's not so important. The next thing to know for the origin of life is this. It's almost certain that the first living molecule was RNA and not DNA. And there's an idea that's called the RNA world, which is a world that consists of RNAs, populations of RNAs that can replicate themselves. And those are not exactly living, but it's really close. Why RNA? Well, first of all, RNA, in addition to being able to store genes, RNA stores the genes of viruses today. So RNA can be a genetic material, but it also can do things. DNA is just a library. All DNA can do is store information, but RNA can act upon the world. Ribosomes are made of RNA. They synthesize proteins. So these are the arguments for why RNA is more likely than DNA to have been the first genetic molecule. And that's pretty much all you need to know. You need to know like why RNA as opposed to DNA. And it's because RNA is single-stranded, it can form these complex shapes, and that makes it catalytic. So that's the basic idea. I have a couple of videos here. But again, here's one thing that I really want to emphasize. If you're feeling stuck, you don't have to suffer alone, and you don't just have to be like going to chat GPT. Because I'm a biology person, I always have a tendency to want to say TP as opposed to PT. Chat GPT, like Chat GPT is wonderful. I use it, but use learn-biology.com. And again, it's like, it costs you nothing, $24.95. Just like talk to your parents. You don't have to call them. I was doing like the universal call sign. It's like, bring your parent over and say, hey, mom, dad, for $24.95, I can use this website. It's really going to help me on the AP Bio exam. It'll teach me all the things I don't know. So there's that. Okay, back to the chat. Natalie, I hope that was useful about the origin of life in the RNA world hypothesis. Looking at the chat. Okay, did that. Osmoregulation of freshwater fish and saltwater. saltwater, freshwater fish. Hannah Lynn. Hannah Lynn, first of all, welcome. It's really great to have you here tonight. And I'm seeing all these other people who I'm really starting to know through this process. That's really fun. So could I talk about freshwater fish and saltwater, saltwater fish and freshwater? I don't think I have a great question. I don't think I have a great picture of that. And I'm a very visual teacher. So, so you know what we're going to do instead? We're going to do something that's similar to that. I'm going to jump back to my slideshow. Okay. I put together a couple of FRQs for us to practice with tonight, and I grabbed them from Learn-Biology, which has a whole library of FRQs for you to practice with. I'm just going to try and find where I put them. Let's go like this. Let's go like this. Let's go like, no, way too far. Way too far. Fabulous. Okay, here we are. Okay. This question, Hannah, is related to your question. And again, one thing I want to emphasize is like this question is from learn-biology.com. So really what I'm hoping is like with all of this participation, I'm hoping like 5 of you at least wind up calling your parents over and saying, hey, mom, dad, $24.95. I'm going to cancel right after the AP Bio exam. You know, we blow $24.95 all the time when we get 2 burritos, stuff like that. So here's a question that I want you to grapple with. And Natalie, you are welcome. So there's a freshwater unicellular eukaryote, and Hannah, I'm going to connect this to the fish thing, has a contractile vacuole that adapts to changes in osmolarity by adjusting pumping frequency. If paramecia need to pump out more water from the cell, the contractile vacuole pumps at a faster rate, and the reverse is true as well. So you can probably see this. This structure over here, that's the contractile vacuole. It contracts and pumps water out of the cell. A student does an experiment where she creates solutions of increasing osmolarity, adds paramecia, and then counts the number of contractile vacuole pumps per minute. None of the solutions have a higher osmolarity than the osmolarity of the paramecia's cytosol. Predict what will happen and justify your prediction using terms related to osmosis. Okay, so basically you're gonna do this. As the solution becomes higher osmolarity, what's going to happen to the frequency of contraction of this contractile vacuole? And I just want to say this question modeled on an FRQ, a short FRQ from previous years. So, so here, maybe I'll just put this in the chat. Of contractile vacuole contraction will, and this is because. Okay, so if you look in the bottom of the comment stream, you can see a comment that I just put in there. And let's see who can take that sentence frame and respond with a sentence that would explain what's going on. Okay, so I hope people are furiously typing, and I hope to see some responses. This is actually new territory for me because I haven't done FRQ response ever on these YouTube lives. Okay, anybody? This is going to take some bravery to do this. I'm going to wait about 10 more seconds. Hopefully I'll see something in the comment stream. Maybe this is too much. Maybe this is too much to ask for students on a YouTube live. But maybe someone's going to surprise me and put a sterling answer into the comment stream. And I'm not seeing anything though. Oh, no, no, no. Okay. Okay. Natalie, first of all, thank you so much for taking a risk. Really appreciate this. So, the contractile vacuole is pumping water out of the paramecium into the solution. Now, here's what I want you to think about, and this relates to Hannah's question. A fish that's in fresh water has a problem. And the problem is that the fresh water, the stream, the lake, is almost always going to be hypotonic to the tissues of the fish. So that means water will enter the fish by osmosis. So freshwater fish constantly need to be pumping out water. And the way that they do that often is by secreting a very dilute urine. So the fish are basically urinating all the time. And that is helping them sort of fight this water that's coming into their bodies all the time. Now, a saltwater fish has the opposite problem. And basically, the saltwater fish is in a situation where water is leaving its body tissues and entering the ocean water. So often, what saltwater fish need to do is they need to excrete salt. And that's sort of how they solve the problem. So, Hannah, I hope that was healthy. Helpful is what I meant. And let's see the answer to this question. Okay. So, prediction. This would have been a correct response. As the osmolarity of the paramecium's environment increases, the rate of contraction of the contractile vacuole will decrease. Why? Because less water is going to be moving in as the solute concentration outside approaches the solute concentration of the paramecia's own cytoplasm. Just imagine this. If this solute concentration were equal to this, then we'd have an isotonic situation. Water would be entering and leaving the paramecium at the same rate. It wouldn't need to pump at all. So at the isotonic point, the pumping falls to zero. As the solution becomes more and more hypertonic, the level of pumping decreases. And I think that's basically what I said. A paramecium cytosol is hypertonic to the paramecium's freshwater environment. As a result, water constantly flows into the paramecium to maintain its osmotic balance or to osmoregulate. The paramecium uses its contractile vacuole to pump water out. As the experimenter increases the molarity of the surrounding solution, the osmolarity of that solution will become increasingly close to the osmolarity of the paramecium. With a water concentration gradient that's less steep, water will diffuse at a slower rate, and the contractile vacuole won't have to pump as often. Eventually, the environmental osmolarity will rise to a point where the environment is isotonic to the cytosol, and at that point, the contractile vacuole wouldn't have to contract at all. So, Hannah, this is basically your question, just with a different organism. Okay, so I really hope that that was hopeful. So I'm gonna be, let's see, let me look at some other questions. I see pragna and low pragna and oh, MHMD. Hey, Mr. W, I study and understand units, but I get confused when answering questions. MHMD. The only thing I can say to you is I've got a solution for you. And the solution is to practice and get feedback. And that's what happens on learn-biology.com. You'll answer questions and you just kind of have to stay cool. You have to stay cool. You have to maintain your equanimity. And you're going to be like, oh, I don't know this, but I think this is the best answer. You click one of the answers and then you see what feedback I've gotten for you. I might just say, hey, MHMD, way to go. Or I might say, no, here's why. And then you study the hint, and that's how you'll improve. I want to emphasize to everybody here that there is still a tremendous amount of time for you to improve your performance, but you have to study in the right way. And there are two ways to do that. One is to go to the College Board's website and do some FRQs. And the other is to practice as many multiple choice questions. And I've got 470 for you. So again, what I'm hoping is that all of you folks who have been here for the first time and like you're asking these questions, they're fantastic questions, but you don't want to rely on me. What you want to do is you want to go through a process where you're finding your gaps and filling them. And the best way to fill them is through learn-biology.com. And it's the best bargain in AP Bio exam preparation. You really, really can't go wrong. I mean, the worst, I guess, is that you'd be out $24.95. Excuse me, my plug just came out. And even though I have plenty of battery, just want to keep things plugged in while we're on Zoom. Okay, so anybody, I think, so I'm looking at questions. Okay, Shyamala Sastry, would it be possible to discuss positive, negative controls and the purpose of controls on FRQs? Okay, so let's see what's a good way for me to do that. I'm going to go back to learn-biology.com. Go there. Okay, so about 2.0. So, Shyamala, the thing for you to do is to go to Unit 4. Okay, Unit 4 is kind of a messy unit in terms of organization, but you'll go to Unit 4, and then you'll go to homeostasis and regulation feedback loops. That's where you'll want to go. And here are the things that I want to emphasize. Like, if you just think about like how your home thermostat works, it's a negative feedback system. And what does that mean? That means that the output of the system winds up turning the system off. That's the basis of homeostasis. Some system gets activated, and then the system's output turns it off. And that can happen in dual loops, where, like, for example, the output of the system, whoa, sorry about that. Here we go. Where the output of the system, like, for example, the heater will turn off the heater, right? The heater will turn off the heater because it'll warm the room in the same way as the air conditioner will turn off the air conditioner because it'll cool the room. So that's the essence of negative feedback. Most of the questions that you're going to deal with related to Unit 4 are negative feedback kinds of questions, but there's also some positive feedback questions. And to get to that, I need to jump a little bit. Oh, right here. So this is a positive feedback system. This is when women are late in labor, what happens is, let's see if I can make this be a little bit bigger. That's good. Okay. So when it's late in labor, what happens is that there are stretch receptors in a woman's uterus. That's what's holding the fetus. And those stretch receptors, they respond to stretch by sending a message to the brain. The brain has a gland that's called the pituitary gland. the pituitary gland releases a hormone called oxytocin, and that oxytocin releases more, causes more uterine contractions. More uterine contractions get picked up by the sensors, and they release more oxytocin. So that's a positive feedback loop. So that's basically like a positive feedback loop versus a negative feedback loop. And Shyamala, I totally messed up because you were asking me about experiments. And you know what? We're going to have to do that in our next session since we're pretty much out of time. But I promise you that I will. Promise you that I will show up. And let's talk about the next session. Let's go to this live reviews. Okay, so the next YouTube live, wow, Saturday night. Did I really commit to doing that? I did. All right, I'll be here Saturday night, 7 p.m. And then I'm going to be here on Sunday. And then I'll be here on the 14th and the 15th. So I'll keep on reviewing with you. But really what I'd like is in return, I don't know, like in return, that's kind of a silly thing to say, but really here's what I'd like you to do. I'd like you to go to this page. So you just type in learn-biology.com, you click student, and I'd like you to sign up for a subscription for learn-biology.com. Every question that folks were asking me tonight would have been answered through learn-biology.com. And what do you need to be doing over the weekend? You need to be practicing. You need to be practicing with FRQs. You need to be practicing with multiple choice questions. And learn-biology.com is the way to do it. It's why I wrote it. I wrote it for my own students because I was sick of textbooks because they weren't learning too much for textbooks. And instead, I had them use learn-biology.com and I got much better results. So look, please do this. Hit the back button. No, that's not what I want you to do. What I want you to do is, where were we? We were going to do this. No, not there. I'm sure you're thinking, wow, this is really boring. I'm just looking for the slide that matters. And the slide that matters to me is this one. Just go to learn-biology.com/apbiology. Ask your parents right now, say, hey, mom, dad, $24.95. It's got tutorials, quizzes, interactive diagrams, games, comprehensive AP Bio exam reviews. Oh, and I forgot to tell you, I also have an app called Biomania. It comes for free once you get your subscription. So don't wait. Like, there's no time to wait anymore. Tonight's the night to really start to do this. So go to learn-biology.com/apbiology. Sign up. Sign up. You won't regret it. It was great being here with you tonight. I really look forward to the next session. And thank you, everybody. And Aiden, thank you. Lily, thank you. Shyamala, thank you. Everybody, please make sure that you've subscribed to learn-biology.com. Quinn, very nice to see you again. Okay. All right, folks, everybody be well. See you at the next one of these. Take it easy.